away we go. Okay, now as I recall, we want to start with looking at nerve impulse conduction. We've talked about how neurons are constructed, and their membrane will start out at a resting potential, just like happens with muscle cells. All right, so their resting potential in the case of a neuron is going to be a negative 70 millivolts. Now, voltage, of course, is like pressure to an electrical current, like pressure is, let's say, for a liquid or a gas. And if it's negative 70, then it's more negative really inside the cell than outside. Now, why? How do we account for that? Well, obviously because of the way in which ions get distributed across the membrane. Remember that the membrane is the lipid bilayer, phospholipid bilayer. Ions aren't going to directly get through that because they're obviously surrounded with water molecules. They're going to have ion channels to let them in and out. Well, there are two kinds of ion channels here. There are transmembrane proteins that serve as sodium ion channels that will let sodium ions into the cell because sodium is present in much higher concentrations outside of the cell than inside. And there are accompanying those potassium ion channels, which will only admit potassium ions, but potassium is in a much greater concentration inside the cell than outside. All right, so in order to maintain this resting potential of minus 70 millivolts, we need to have the sodium staying out and the potassium staying in. But what's the charge on a sodium ion and what's the charge on a potassium ion? Well, they're both plus 1, right? All right, so obviously that's not going to account for the negative voltage difference. What is inside the cell that accounts for most of that charge is going to be the proteins. Proteins at normal pH, not only in the blood, tissue fluids, and in the cells, at normal physiologic pH, proteins have a negative charge. And there are a lot of proteins in the cytoplasm of the neuron. That is where we get the negative charge, all right, compared to the inside versus outside. And to maintain it, we have to have, of course, that distribution of sodiums and potassiums that we talked about. All right, so a neuron membrane, which has its resting potential, is said to be polarized. Polarized having a charge crossing. All right, now, we've already said, we have these differences in ions. I wouldn't worry about this, but we'll just mention it. There's typically like 14 times the sodium on the outside as on the inside, and there's about 30 times the potassium concentration inside the membrane than on the outside. But in any event, there we go. Now, the main reason, as we said, why it has this negative 70 millivolt resting potential is because of the proteins and the distribution of the sodiums and the potassiums. All right? And so, there we go. And I don't see the sense of belaboring that point. Now, during rest, there is a tendency for potassiums to want to escape. To leak out through some of those potassium ion channels. All right, well, there's a way to pull them back. Because for one thing, the negative charge on the proteins wants to pull them back in, right? And that helps control how they can leak out. And the potassium ion channels are not freely open, so they're not going to be able to gush out anyway. And maybe a few sodiums like to leak in. Well, let them. Don't worry about it. How are we going to maintain the resting potential of minus 70 millivolts? Well, one of the things that we're going to find in here is that we have a sodium-potassium ion pump. All right? Another transmembrane protein that you find embedded in these membranes is this sodium-potassium pump. Now, what does it do? Well, as the name implies, it pumps sodium ions out of the cell, because they don't belong in there, they need to be outside. And it pumps potassium ions in the opposite direction into the cell, because that's where we want them. Remember, sodium is the major extracellular cation. Potassium is the major intracellular cation. Alright? So, sodium's leak in, we want to pump them out. Potassium's leak out, we want to pump them back in. Sodium-potassium pump does that. And it helps to maintain this resting potential of minus 70 millivolts across the membrane. Now, it is a type of active transport. Sodium-potassium pump is a type of active transport. But what fuels active transport mechanisms? ATP, right. All right. Now, simple enough. So the sodium-potassium pump is going to help maintain the resting potential. But it's also going to help out in a little bit when we generate an action potential too. All right, so there we go. We've talked about, active transport, and just like with other active transport mechanisms, this one is taking the sodium and the potassium both from areas of low to high concentration. Okay? Now, when it's time to get a nerve impulse started, what we can call an action potential. What happens? We have to apply a stimulus to a section of membrane. All right? How do you apply the stimulus? Well, there are three ways to do it. And we'll see that on a later slide, but I'm going to mention it right now. Number one, it can be chemically delivered, a chemical stimulus. All right, now, what kind of things provide a chemical stimulus to generate an action potential on a section of neuron membrane? Well, neurotransmitter substances can do it sometimes. All right, and remember our famous neurotransmitter substance when we were talking about neuromuscular junctions. Which one was it? Acetylcholine. All right, that can do it. Another way that you can generate an action potential on a section of membrane is to alter the temperature. A temperature change on a section of membrane might do it. Or another way of getting a threshold stimulus on a section of membrane is pressure. Some kind of pressure applied to a section of membrane. It doesn't matter which of the three you use. They all three are going to cause the same thing to occur. What is that? They're going to cause sodium ion channels in that section of the membrane to open up and let more sodium come gushing into the cell. All right? Just remember now, outside the cell, lots of sodium, not very much inside. If a sodium ion channel opens up, we'll freely let sodium ions through, and those sodium ions want to go from an area of high to low concentration. So you open up the sodium ion channels a little, and the sodium ions start to come in. Now, when they start entering the cytoplasm, what is going to happen? Well, that section of the membrane is going to start to depolarize. So, opening sodium ion channels will begin the process of depolarization of a section of neuron membrane. All right? So, initially, we let in enough sodiums, and it goes from being negative 70 millivolts, that's the resting potential. It increases initially to a negative 60 millivolts, and we reach the threshold level. Now, what next happens, those sodium ion channels that were opened up a little bit pop open completely, and sodium just gushes in to that section of the cytoplasm. All right? So what's happening now to the sodium ion concentration in the cytoplasm in that little area of the cell? It's going very high. All right? And as it does, as it goes higher and higher and higher, that section of membrane becomes more and more positive. You know, minus 60 is more positive than minus 70, right? 0 is more positive than minus 70. And it doesn't stop there. It'll actually continue on to plus 30 millivolts. All right? And it may go just a little higher than that, but plus 30 in that section. Just that section. All right? Now, when we get to plus 30 millivolts on that section of the membrane, we would say that that section of the membrane is fully depolarized. Because remember now we talked about muscles. When you depolarize the membrane, it changes from a negative charge to a positive charge, right? All right, well, that's the same thing here. That section of membrane that is now fully depolarized has gone from its negative 70 resting potential up through 0, all the way up to, say, plus 30 millivolts. But what about the areas that are adjacent to that? They haven't changed. They're still at resting potential for now. All right? Because they're not going to stay that way long. All right, so what's going to happen as more and more sodium gushes in and changes the charge on that section of depolarized membrane at plus 30 millivolts, there's now going to be enough sodium in that cytoplasm in those adjacent areas to cause the sodium ion channels there to pop open as well. So what's going to happen in those areas? They're going to start to depolarize as well. All right? Now, guess what's going to happen? We're going to get propagation. We're going to get spread of that depolarization from where it started, that one little section, all the way across the membrane and down to the very end of the axon. And how many axons do you have on a neuron? Just one. Right. All right, now, we're not done. Because obviously, if we want this neuron to conduct another impulse, and we do, we want to use it over and over again, we're going to have to repolarize the depolarized sections. Well, let's go back to the original section. All right? What caused the depolarization to occur there? What did we open up to cause that to occur? Sodium ion channels. All right, once we get to that plus 30 millivolts, total, or complete depolarization, basically, the sodium ion channels are now going to close. No more sodium is going to be admitted at that area. All right, but now what is the charge across the membrane? It's still plus 30, right? Hasn't changed. We just aren't letting any more sodiums in, so it isn't going to go any higher, but we haven't done anything to change it. It's still plus 30. Well, now that's going to change. Because when we get to that plus 30 millivolts of a fully depolarized section of membrane, the potassium ion channels, remember those? The potassium ion channels are now going to pop open. But what's coming out? Potassium. And potassium ions now, which are in a much higher concentration inside the cell than outside, are now going to diffuse out. All right, so the potassium ion concentration in that section is now going to be very high on the outside. Now, again, what was the charge on a potassium ion? Plus 1. All right? We put a bunch of plus 1s in. Now we're bringing a bunch of plus 1s out. What is going to happen to the charge on the membrane as more and more potassium exits the cell? It's going to become more and more negative. That's exactly right. And ultimately, there's going to be enough potassium exiting to bring the charge back to the resting potential of negative 70 millivolts. All right? And then, what's going to happen with those potassium ion channels once we get back to negative 70 millivolts, I wonder? It's going to close them up. They're going to close up. All right, yeah, we're ready to start over just yet, but we'll get there. All right, now we are back to negative 70 millivolts. We're now back to the resting potential in that area. Now remember, the depolarization has continued to spread away from that. So we've got sections that are depolarized and sections that are in the process of depolarizing and sections that haven't yet depolarized, and then back where we started, they're repolarizing, and that's going to spread too. All right? But let's go back to that original section. All right, it's repolarized. It's back to negative 70 millivolts. But where are the sodiums and where are the potassiums in that section? Yeah, they're in opposite position from where they were originally. We've got a lot of sodium inside and a lot of potassium outside. Now, it's repolarized, but if we want to be capable of depolarizing that section of membrane again, we're going to have to move the sodiums and the potassiums back to their, what I like to call, starting positions. So we've got the sodium out and the potassium back in. Now how are we going to do it? Well, we've already got the means at our disposal. What was that third transmembrane protein that we said that was embedded in the membrane in that area? The sodium-potassium pump. So guess what it's going to do? It's going to pump the sodiums out. It's going to pump the potassiums back in. And once we get them back to their starting position, and we still have this minus 70 millivolts, but now there's lots of sodiums out, lots of potassiums in, you come along and apply another stimulus to that section of membrane, what happens? You generate another action potential. There you go. Now what can be simpler than that? Not much of anything. Alright, the sodiums and potassiums change positions, but we're going to bring them back to where they were originally. And when they're back where they were originally, we can conduct another impulse. You can't do it though until you move them back. Alright? Now, there we go. Alright, so we now have more sodiums, as we said, coming in. That sodium potassium pump can temporarily pump out. And that's what changes, of course, the charge. And so now, there we are, and it's fully depolarized. And to get it repolarized, we have to open up the potassium ion channels, and they move out. And now, there we go. And I'm not going to worry about it because it's all here what I said. And you can sort it out later. All right, so there we go. So, in order to get them back to their starting positions, we can rely on the sodium-potassium pump. What was it? We said we had to have fluid, ATP, right? Okay, one iteration. And so now, how long does it take? Oh, half a millisecond or so to get it repolarized. It works pretty fast, really. All right, now, once the membrane has been restored to its resting potential, it can once again conduct another impulse. We can generate another impulse, or, as we might call it, action potential. Now, the period of time required for the membrane to regain its resting potential and to have the sodiums and potassiums fully restored to their starting positions is the refractory period. In fact, what we would call the absolute refractory period. All right? Absolute refractory period is how long it takes after an action potential is passed to bring us back to the resting potential and have the sodiums and potassiums fully restored to their starting position. Now, there is something else called a relative refractory period. And I'm going to skip down to it, and then I'll go back to that other thing. All right, relative, well, what's that mean? Well, it means that we're back to a resting potential, minus 70 millivolts. And we've started pumping sodiums out and potassiums back in. but we haven't fully restored the sodiums and potassiums to their starting positions. All right? There's still more sodium to pump out. There's still more potassium to pump back in. But when we're in that relative refractory period, we can still get an action potential generated on that section of membrane. But what is it going to take to do it? It's going to take something greater than a threshold stimulus to do it. Now remember, what did the threshold have to do? What did it have to open up? It had to open up enough sodium channels to admit enough sodium to depolarize the membrane. All right? Well, if you haven't got all the sodiums pumped back out, you're going to have to add even extra sodiums if you want to generate an action potential during that relative refractory period. It can be done, but it takes a stronger stimulus that opens more sodium channels and emits more sodium than otherwise would be the case. Which is longer, which is shorter? A relative refractory period or an absolute refractory period? Which one's shorter? Which one has a shorter duration? Relative, yes. Because you don't have to have it all totally reset. You don't have to have all the sodiums pumped out and all the potassiums pumped back in. Okay, we're on our way, we're not quite there. If you apply a stronger stimulus, you can get an impulse. Absolute refractory period takes a little bit longer. Why? Well, it takes some time to pump those sodiums out and those potassiums back in. All right? There you go. That's the difference. That's the difference. Think about it. All right, now let's go back and take a look at this. The size of the fiber, the diameter of the fiber, has an influence also on how long it takes to have a refractory period, so you can transmit another impulse. Larger fibers tend to repolarize faster than the smaller diameter ones do. I wonder why. Well, let's just think. What caused the action potential to start in the first place? Why'd you have to open up? Sodium channels. Sodium gushes in. All right? Now, if you've got a really large diameter fiber, what about looking at the sodium concentration in cross-section there? Where's more of the sodium going to be found? Near the surface of the membrane, down the middle of the cell? Closer to the surface, isn't it? Right? Isn't it? Well, what about diffusion? What is it? Going from here, high to low concentration, right? Well, if sodium is high outside, low inside, as it comes in, it's going to be a lot higher near that membrane, that inner surface of the membrane, isn't it? Than it is down there in the center. All right? What about one that isn't very large in diameter? Well, yeah, there's going to be more near that membrane, inner membrane surface, but it's going to be pretty well filled up with sodium down there, isn't there? Well, relatively speaking, it's going to take longer to pump that out than it is when it's just up there near the membrane. Right? Think about it. All right. All right, so there we go. Now, let's just take a look at this little diagram. This is kind of, again, showing us the resting potential for the membrane. All right, so here we are. Cross-section of membrane, actually longitudinal section. There are the two membrane surfaces, okay? Because remember, it's a tube, essentially. All right, there are sodium-ion channels. There are potassium-ion channels. And there's the sodium-potassium pump. And they're all examples of transmembrane proteins. Okay? All right, now, when this is resting, what's happening? Well, some sodium, a little bit, might get in. All right? A little bit, not much. Sodium-ion channels are pretty good about keeping sodium out. All right? Potassiums are a little leakier. More potassium diffuses out. All right? Whoopee. What's going to keep them in line? that sodium-potassium pump. Pumps the sodiums out, pumps the potassiums back in, maintains a negative 70 millivolt resting potential. There it is at rest, right? Now, what's going to happen when we get ready to conduct an impulse, to get a section of membrane to depolarize? Well, we apply to that section of membrane either a chemical stimulus, a temperature difference stimulus, or a pressure stimulus. And what does it cause to open up? Those sodium ion channels open. And sodium comes gushing in. All right? So what happens to the sodium concentration in this area of the cytoplasm? It's going up. The sodium concentration here and here is going down, right? And what's happening to the charge from inside to outside across that membrane? It's becoming more positive as more sodiums enter. All right? Remember, very negative in here because of all the proteins. But if we keep adding more positively charged sodium ions to this, to go with the potassiums that are already in there, it's obviously going to become more positive. All right? So, what happens? The charge starts to increase. It goes initially from minus 70 to minus 60. The sodium channels really pop open. The sodium gushes in. And the charge goes on up to 0 and then on up to plus 30. And when it gets to plus 30, these close shut. And these things pop open. Now, what happens with potassium? It floods out. And what happens to the charge on this section of membrane? I'm going to get my fancy new pointer. What happens on this section of membrane now as potassium diffuses out? It starts to become more negative again, doesn't it? Right? So, it started out at plus 30. Well, now it's going to go to 0, and then it's going to keep going until we're down to negative 70, and we repolarize that section of membrane. All right? Simple. But what's happening down here and over here? Well, all that sodium that entered here doesn't just stay here. It diffuses off these adjacent areas, doesn't it? And guess what's waiting on it when it gets to those adjacent areas? More sodium ion channels. What are they going to do? They're going to open up and let more sodium in that area. So the sodium, in essence, is getting higher not only here, but it's also going to the adjacent areas. Now, they were originally resting potential, but they're not going to be for long when you open the sodium channels. And then, when they fully depolarize, what's going to happen with potassium ion channels? They're going to open up, and it repolarizes. Now, what do we have to do to get back through that refractory period? Well, what do you suppose the sodium-potassium pumps are going to do? They're not potted plants. They're not just sitting there looking pretty. They're going to spring into action. They always are active to some extent. But now they're really going to spring into action. What are they going to do? Pump that sodium out and pump that potassium back in. And what's eventually going to happen? Well, negative 70 is still maintained, but we're back to the original starting positions. Lots of sodium on the outside, lots of potassium inside. And now, if we come along and boink that with another stimulus, what's going to happen? Another action potential is going to be generated that propagates, that spreads down the membrane. All right, so here we are in another little diagram. Negative 70 millivolt resting potential. There we are, sodium's on the outside, potassium's on the inside. All right, apply a stimulus, open the sodium channels, in they come. What happens? We go from negative 70 to negative 60 to 0 up to plus 30. And then Sodium channels close. Potassium channels open. Here we go. Sodium's inside. Potassium comes out. What happens to the charge on the membrane? Drops right back to -70. Now what can be simpler than that? Now how are we going to get back to this? How are we going to get from here to here? Maintain that -70 millivolt charge on that section of membrane. We're going to reset the Bedoin plot, firing up that sodium-potassium pump and pumping them sodiums out, and the potassium's back in, and there we are right back to that. And now we're ready for another action potential. Alright? So when everything's totally reset, we've gone through the absolute refractory period, and we're all back to the starting point. What would be the difference between this and a relative refractory period? Now, relative refractory period, yeah, we're at -70 millivolts, but what about the positions of those ions? They're still in the process of being sorted out and moving, right. We don't have all the sodiums out, and we don't have all the potassiums back in. Alright? We've gotten to a place where there's most of the sodium is out, and most of the potassium's back in, but it isn't complete. And so if you wanted to generate an action potential under those conditions, what do you have to do? You have to give it a stronger than threshold stimulus that opens even more sodium ion channels. Okay? Now that's simple enough, isn't it? Heavens, yes. Well, let's look at this next thing. Alright, and I don't like that. Already said that. Alright, here we go. Alright, here we start with an action potential. There's the sodium coming in. Alright, what happens to the charge here? It went from -70 to what? Huh? Well, it goes ultimately up to +30. Alright, and then we repolarize, so this section is now reset to -70. But what about this section? It's now depolarizing, right? Alright, the impulse is passing from one section to another. That was depolarized, that was repolarized, that's still at resting potential. And when we come to the next one, what's happened? We've now depolarized the next section. The prior sections have repolarized back to the resting potential. And this one's probably now moved the sodiums and potassiums back to their starting position. Oh, it's simple. It just propagates. It spreads from one section to another. As that sodium concentration, that high sodium concentration, diffuses down the cytoplasm of the exoplasm. Alright? Alright, so let's take a look at this little summary table. I don't think this really printed out in the outline, but you can go get it. But I mean, this is the same stuff we've already said. Alright, so the nerve cell membrane maintains the resting potential by diffusion of sodium and potassium down their concentration gradients as the cell pumps them out to maintain the gradients. And what maintains the gradient? The sodium-potassium pump. Alright, neurons receive stimulation, causing the local potentials, which may some to reach threshold. What were the stimuli that would open up those sodium channels? Chemical, temperature change, or pressure. Alright. Sodium channels in the local region open up. Sodium ions diffuse inward, depolarizing the membrane. What was the resting potential originally? -70 millivolts. What happens when it's fully depolarized? What does it go to? +30 millivolts. When that reaches, potassium channels open. Potassium starts to come out. What happens to the membrane? It starts to repolarize. Potassium ions diffuse outward, repolarizing the membrane. The resulting action potential causes a current that stimulates the adjacent portions of the membrane. The action potential spreads or propagates. and a series of action potentials occurs sequentially along the length of the axon as the nerve impulse. All right, there you go. So, if this depolarization starts, where usually on a dendrite of the cell body, but remember it always spreads to the axon, and once it gets there, it goes the entire length of the axon. So if this starts, it propagates, it spreads. Now, in order to get it to do that, you do have to apply a threshold stimulus, a stimulus of a certain magnitude that opens enough sodium ion channels. What would happen if you just applied a subthreshold stimulus to a section of membrane, and you only opened a few sodium ion channels, and you only let a little bit of sodium ion come in? Now that's going to happen. Why? Because the sodium potassium pump is going to pump it right back out again, very quickly. All right? It wants to maintain that resting potential. The only way you're going to get an action potential is if you let so much sodium gush in an area that you overpower, if you will, the sodium potassium pump. It's coming in faster than it can pump it out. All right, just like in the Titanic, the water was coming in faster than the pumps could take it out. What happened to the Titanic? It sunk. That's right. Well, kind of the same difference. All right? Only in this case, it's sodium ions. Well, that's simple, isn't it? Good heavens, yes. All right, another wrinkle on the proof, and then we'll get to the animation you'll love, because this can save probably a minute or two of what it's taking all this time to blather at. All right. Conduction along axons. There's two ways to do it. It can either be continuous or it can be saltatory. Now, where do we find which one? Continuous conduction is what you will observe with non-myelinated axons. So if they're non-myelinated axons, or what we could call gray fibers, right, they will only show continuous conduction. Myelinated fibers, or white fibers, right, because myelin's white, those will demonstrate what's called saltatory conduction. Now, what's the difference, and why do we care? Well, as we will see, continuous conduction conducts slower than saltatory conduction, because myelinated fibers are going to conduct much, much faster. All right, now, continuous along the entire length of the axon. All right? Now, if you're going down the axon and you've got continuous conduction, you have to depolarize every section along that membrane, which means you're going to have to open and close every single solitary sodium ion channel as you pass down that membrane. Takes a little time. Saltatory conduction, the only place the membrane will actually undergo depolarization, repolarization, the only place where you'll be opening and closing sodium and potassium ion channels, at the nodes of Ranvier. That's it. That's it. It doesn't take nearly as long to conduct the action potential down a fiber if all you have to do is open and close those at the nodes. All right? Now, why aren't they opening and closing all the way along? Well, they're covered up with something. What are they covered up with? Myelin. They can't open and close. They're clamped shut because of that myelin. And you're maybe thinking, well, how on earth can the depolarization jump from node to node to node? Well, when they open, what's coming in? When the sodium ion channels open up in an area, what's coming in the cell? Sodium ions. Does that sodium ion just stay where it entered? No. It diffuses down the axoplasm, and when it reaches the next node, those sodium ion channels are going to pop open, all right? And more sodium rushes in and continues to diffuse on down to the next node. They open up, and when they open up, those areas are depolarizing, right? Well, isn't that simple? Yes. We'll show you a little animation of that here in just a very short while. All right? And let's just take a look at this for comparison's sake. And I wouldn't worry about those numbers, but I just want to use them for illustrative purposes. Myelinated fibers will conduct an impulse at the rate of something like 120 meters per second. Do you have 120 meters of axons going even to the muscles down in your lower leg? I doubt it. Myelinated fibers very fast. Unmyelinated much slower. What, about half a meter per second? So, guess what kind of fibers you want controlling your muscles, your skeletal muscles? Probably myelinated fibers, I think. You know, if you want to have nice, quick reflexes, you probably want to involve myelinated fibers in that. Now, what about the GI tract? You know, even if you need a little hypermotility, you can go with unmyelinated, very lightly myelinated fibers there, right? Now, see, isn't that simple? All right, I think, okay, let's take a look at this. Here we have two Schwann cells. There is an area between the Schwann cells, between the adjacent Schwann cells, where there's a little break in the myelin. There's no myelin there. What's that thing called? Node of Ranvier, right? Now, if we were sending a nerve impulse down this fiber, that's the only place the sodium and potassium channels are going to pop open and close. That's the only place where the membrane really depolarizes and repolarizes. Sodium, potassium channels aren't going to open here and here. Why? They're covered up. But sodium concentrations here in this axoplasm, high, it diffuses along here, and it gets even higher here, and it just keeps diffusing until we get to the end. All right? Well, that's simple, isn't it? Yes. All right. Well, we'll come back to synaptic transmission in a bit. I think we'll look at the animation, and I think it'll look better probably on the recording if I cut these lights before we go each way. And I think I've got the sound on, so let's minimize this, and we'll take a look at the animation. So let's just see what we've got here. And the one thing about this, it has some really awful, I think, irritating background music, which I wish they'd left out, but hey. Away we go.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon. This multimedia presentation will be most helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon. Here is the neuron, and you can see the repeated Schwann cell membrane forming the myelin. Note that there is a small space between the Schwann cells where the axon is not covered by the neuroglial cell. These spaces are called nodes of Ranvier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another. An action potential starts on a polarized membrane, which is -70. A stimulus causes the sodium gates to open slightly, and sodium starts to trickle into the cell. If the cell reaches -60 or threshold, the sodium gates open wide and sodium floods in, bringing the inside of the axon to +30. At this point, the sodium gates close and potassium gates open. Potassium starts to pour out of the cell. This allows the neuron to become polarized again. Then the sodium-potassium pump starts to actively transport sodium out and potassium back into the neuron. First, we will look at the propagation of the action potential in the unmyelinated axon. Propagation is the repeating of action potentials down the axon. The action potential is repeated because as the sodium comes in, it diffuses to adjacent areas within the axon. As the sodium increases in this area, threshold is reached. Sodium gates open wide, sodium rushes in, causing depolarization and an action potential. As the sodium enters this area, it diffuses through the axoplasm and another action potential is created. This continues down the length of the axon. Now look at the myelinated axon. The same process applies to the myelinated axon. An action potential develops, and as the sodium comes in, it diffuses through the cytoplasm of the axon. It continues to diffuse through the portion of the axon wrapped in myelin. The increased sodium concentration reaches the node of Ranvier, increases the axoplasm to -60, and depolarization occurs. The sodium gates open wide, sodium floods in, and we have an action potential. Again, the sodiums diffuse through the axoplasm, reaching the next node. An action potential develops. The process is continued down the myelinated axon, passing from node to node. Compare the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
Now, simplifies it, doesn't it? Well, one more, and that's the sodium-potassium pump. So, let's get to that. And away we go.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Now, cleverness, Paul. All right. There we go. Okay, so back to my sleeping pill. All right, now, synaptic transmission. Now, we've already had a foretaste of this one, too. Remember with muscles, how did we stimulate a muscle to contract? We're thinking about skeletal muscles. They have a neuromuscular junction, right? All right. Well, basically what they have is a synapse, and the synapse is a connection between neurons. So this is how an impulse passes from one neuron to the next in a chain of neurons, right? Because remember, we have sensory neurons that pick up impulses from receptors. They relay those, the old action potential business, into the central nervous system, where they're going to synapse with an inner neuron. All right, it's going to then generate an action potential that then will synapse or connect with a motor neuron that then stimulates an effector to operate either a muscle or a gland to respond, right? All right, so here we go. Impulse transmission across the synapse from one neuron to another. And the two neurons that we would have in this little chain of two neurons, one is going to be called the presynaptic neuron. That could be like a yes, my mind just went blank, just like a sensory neuron, all right? And then it's going to pass that information on to a postsynaptic neuron at the synapse, and we have a chemical signal going from one neuron to the other that causes the second one, the postsynaptic one, to generate an action potential and propagate it. All right? All right, so there we go. And so, how are these things put together? Well, very similar to what we've already seen. The axon of the presynaptic neuron terminates in a synaptic knob on the membrane surface of the postsynaptic membrane, or postsynaptic neuron, rather. All right, what's a synaptic knob? Terminal end of what? Teledendria, right, that come from the axon of the presynaptic neuron. All right? What's in those synaptic knobs? What do they contain? Huh? Yeah, you have neurotransmitters packaged up in little synaptic vesicles, right? And when an impulse comes down to that area, we're going to find this neurotransmitter released. And it's going to then stimulate the postsynaptic neuron, or generate, if you will, an action potential in it. All right. So the synaptic knobs are in contact with either the dendrites or the cell body of the postsynaptic neuron. No shock there. Remember the model we had the other day that showed all those synaptic knobs connected to the dendrites of the cell body of the neuron model. All right, in the synaptic knob are the synaptic vesicles that contain neurotransmitter substances like acetylcholine, which is an excitatory type of transmitter. It stimulates the production of an action potential in the next neuron. Now, here we have a neuron, and this would be our postsynaptic neuron, and here we have a teledendrian from a presynaptic neuron. There's the synaptic knob out here on the dendrite. here we have another axon with several telodendria synapsing with the membrane of this postsynaptic neuron right here on the surface of the cell body. So here is our dendrite cell body and the axon of the postsynaptic neuron. Now, impulse comes along here. We're going to release neurotransmitter. What's going to happen at that location on the membrane of this postsynaptic neuron? Generates an action potential, right? Spreads all the way down. What about this one? If you get an impulse here, you're probably going to generate action potential in these areas. Over the cell body, it just spreads down the axon hillock, down the axon, there it is. All right? Simple enough, isn't it? Yes. All right, now, come on. Nerve impulse reaches the synaptic knob. What does it do? Well, one of the things you find in the membrane of these synaptic knobs, just go right back to them for a moment. In their membranes, you're going to find calcium ion channels, another type of transmembrane protein. All right? If an action potential reaches those calcium ion channels, guess what they do? Pop open. And when they open up, they emit calcium ions into the axoplasm in that area. All right? So the calcium is now flooding in to the axon, into that synaptic knob. What does the calcium do? Well, it's going to cause the synaptic vesicles to fuse with the membrane of the synaptic knob. That's going to cause exocytosis, or in other words, an opening of those synaptic vesicles. And what floods out of them? The neurotransmitter substance. Now, where does it go? Well, remember now, there's a little gap between the synaptic knob and the surface of the membrane of that postsynaptic neuron. Remember what that was called when we were looking at the gap between the synaptic knob and the motor end plate part of the muscle cell membrane? What was that little gap called? Synaptic cleft. That's it. Guess what it's going to be called here? Synaptic cleft. All right, there you go. So, the neurotransmitter substance diffuse across the synaptic cleft. They absorb the receptors that are embedded in that membrane surface. All right, again, another transmembrane protein, basically. And the binding of the neurotransmitters is going to do what? It's going to open up the sodium ion channels in that region of the membrane. And if you open up enough sodium ion channels in a region of the membrane on a neuron, what is generated? An action potential, right? What could be simpler? So what's happened? The nerve impulse has gone from a presynaptic neuron to the postsynaptic neuron, transferred over chemically by that neurotransmitter substance. What did the neurotransmitter substance do? Open the sodium ion channels and depolarize that part of the membrane and generated an action potential. Now it's just that simple. All right, so let's take a look at this little diagram. So here it is writ large. This one's nicer, it's bigger, it's easier to see. There's the synaptic knob. Inside it are these synaptic vesicles. What do they contain? Well, the little blue balls would be the neurotransmitter substance like acetylcholine, for example. All right? This part of the membrane has depolarized, and action potential has passed. That has opened the calcium ion channels. Calcium comes in from the extracellular fluid. It then combines with the membrane around these synaptic vesicles, causing them to fuse with the membrane. That causes them to open up. We have exocytosis of the neurotransmitter. It floods into this synaptic cleft. it comes over here, diffusing across and combining with the neurotransmitter receptors that are embedded in the membrane here of our postsynaptic neuron. All right? Binding of the neurotransmitter, the acetylcholine, let's say, causes an opening of the sodium ion channels, and that part of the membrane is now depolarized. What about these adjacent sections? Well, initially they're polarized, right? But what's going to eventually happen to them in very short order as sodium comes flooding in here? They're going to depolarize as well, and that action potential spreads off in both directions. And then this section will repolarize, and then these eventually will too. But the action potential is already passed. Oh, there it is. Pretty simple. Now, do we want to just leave the acetylcholine there forever? Nah. How do you get rid of it? How did you get rid of it before? It's going to be the same thing this time. Acetylcholine esterase is absolutely going to hydrolyze it and break it down into acetic acid and choline, and it's no longer active. Now, if you want to depolarize this membrane section again, how are you going to do it? Release more acetylcholine. That's right. So that one action potential has to be followed by another, right? And another if you wanted to keep transmitting that nerve impulse across the synapse. So here's a synapse. Synaptic knob of a presynaptic neuron and section of the membrane of the postsynaptic neuron. Okay? So where the two neurons interconnect, if you will. And here is an electron micrograph of a synaptic knob in blue, and there in yellow, those are the little synaptic vesicles containing neurotransmitter. And then this little yellow gap here is the synaptic cleft, and this in red would be the membrane of the postsynaptic neuron. And so if an impulse comes along here, opens calcium channels, calcium comes in, combines with these membranes, causes them to fuse with the membrane, opens them up, acetylcholine or whatever the neurotransmitter is they contain, diffuses across the synaptic cleft and combines with receptors that are embedded in that membrane, opening, if you're going to depolarize it, opening the sodium channels. Okay, right? Yes. All right, there we go. Now, here's another wrinkle, though, to the proof. All right, as we note, when it comes to the changes that occur in that membrane of the postsynaptic neuron, some neurotransmitters, like acetylcholine, are excitatory. So what are they going to do? They're going to depolarize that section of the membrane. All right? No problem there. Well, there isn't one here either. Some neurotransmitters, like dopamine, on the other hand, have the opposite effect. They're what are considered to be inhibitory neurotransmitters. Now, what do? Well, they bring about a hyperpolarization of the postsynaptic membrane. Hyperpolarization. What does that mean? Well, now, what is the charge on a polarized membrane? What's the resting potential? Negative 70 millivolts. Hyperpolarized is more negative, like minus 90 millivolts. All right? Now, if you have a hyperpolarized membrane, how are you going to depolarize it? What would you have to do if you wanted to depolarize it? Yes, you'd have to open up even more sodium ion channels and let in even more sodium if you wanted to do that. Chance is pretty high it isn't going to happen. Now, we have a balance going on, and there are bonds between excitatory transmitters like acetylcholine and inhibitory transmitters like dopamine. And it's a balance between the two that will account for our ability to get fine muscle movements. All right, it's a matter of voting. Who wins the election? The person that gets the majority of the votes, right? Or at least a plurality when you got a gazillion candidates, right? Well, here it's a majority because there's only two. If you've got excitatory impulses outnumbering inhibitory ones, what happens? You generate an action potential in the postsynaptic neuron, and an impulse is generated and propagated and conducted. What if, however, there are more inhibitory signals arriving than excitatory ones? No action potential, and nothing happens. Now let's just think. What happens in someone with Parkinson's, where this is out of whack, where they're generating lots of excitatory impulses and not enough inhibitory ones? What happens?
Muscle jerking.
Lots of muscle jerking, the tremors, and and they have a hard time maintaining posture and balance and so on. Well, their muscles are hyperactive, so to speak, because they're getting far too many impulses. It's not being well controlled. And the area where that's happening is up here in those basal nuclei and basal ganglia. There are basal nuclei up here in the brain, and we'll chat more about that next week. But in any event, there you go. Remember when the tracts that the nerves, like the corticospinal things, pass right through that section of the brain? Did you talk about tracts in lab? You will, don't worry. All right. The seeds have been planted. All right, so there we go. All right, now here we have a neuron. This is kind of like the model we had. Look at all those axons coming in here on one poor little neuron. Some of these things, probably bringing in excitatory impulses because they're going to release acetylcholine. Some of them may be inhibitory. What's going to happen if you have more inhibitory release than excitatory release? Is this thing going to generate an action potential? No, it isn't. What if there are more excitatories than inhibitories? Then it does. Neat. All right, impulse processing depends on neuronal pools. Networks of presynaptic and postsynaptic neurons. All right, so a lot of times we find these involved with what we call interneurons, of course. And where do you find the interneurons located? Central nervous system. All right, now pools can either be of two types. They're either converging or diverging. Converging or diverging. Convergence. Axons of more than one presynaptic neuron synapses with a single postsynaptic neuron. All right? Each axon can supply subthreshold stimulus, and the sum of the two might sum the threshold and generate an action potential. Divergence, axon of a single neuron may branch and synapse with more than one postsynaptic neuron. All right, divergence can have the effect of amplifying a response because it can get several postsynaptic neurons to generate an action potential. Here's a diagram. A would be convergence. Look at that. 1 and 2, presynaptic neurons. They're synapsing with a single postsynaptic neuron, number 3. All right? Either one of these supply threshold stimulus, or both of them giving us a sub-threshold stimulus, a sub-threshold, well, that generates an action potential or propagates. All right? Another thing you might find here is one of these may be releasing inhibitory transmitters and the other one is excitatory. What's going to happen if only the inhibitory one is released? Nothing, yes. Now here, there's one presynaptic neuron and two postsynaptic neurons. Look at that. What have we got? We've got axon collaterals and chylodendria there with both. All right, if this generates a threshold stimulus, propagates it, passes it on to those, what happens? They both generate an action potential and transmit. All right? The effect really of amplification. Now, this one might control one set of muscles. This one might control another set. All right? So you can get even more cells contracting in response to the output of that one single lonely important thing. Presynaptic neuron. Well, you've suffered enough. Now, this is where the test stops. And the exam is next Thursday. And then Tuesday, we're going to go into the brain and spinal cord. Mostly likely the brains go. That's what we're going to get. Some of that we talked about in lab, so we can go over some of it a little quicker than others. And you'll enjoy it immensely. And of course, that material will show up on the final exam, stuff that you have not yet been tested, but that's okay. There will be plenty of old things. Well, more about that next Tuesday.